All right, we are moving into meiosis 1. Remember, meiosis is a two-step process. It's known as a reduction division. So we're going to see throughout meiosis 1 that we're going to reduce the number of chromosomes in the cell by a factor of 2. We're going to become haploid. And then in meiosis 2, we will split sister chromatids apart. That's the division part. So right now I'm showing that our DNA is condensed by putting my replicated pairs across from each other here. So I've got my X's of identical information. 2 yellows are sister chromatids. A red and a yellow that are the same size are a homologous pair. They're called a homologous pair because they carry the same information. So for example, if right here I had a gene from my mom for eye color, maybe she gives me the color blue as a trait. This would be the gene from my dad, and he might give me the color blue as well, or maybe he gives me the color brown. Depends on what he's got in his genes, all right? But they carry the same type of information at the same location, what we call loci. So I've got homologous pairs, and when we talk about meiosis 1, you really want to focus on this idea of homologous pairs. That's where your attention should be, okay? So in prophase 1, the DNA condensed. The nuclear membrane would actually disappear, it would dissolve, so I'm going to take that out of there. And something very interesting happens as well, and it is called crossing over. I am not drawing in here the centrioles or the spindle fibers, only because I'm going to have to keep reusing the same cell over and over. As you draw your pictures, you should be drawing in centrioles and spindle fibers. Spindle fibers would be connecting to these homologous pairs. They are not necessarily lined up neatly in the middle yet. Sorry, that was just a coincidence that they looked like they were. The coolest thing about prophase 1 is that crossing over occurs. So in crossing over, because we have these two pieces that have identical information and are very similar chemically, they can swap and we get a little trait of info, something that adds genetic variation to the system, which is a good thing. So we're going to take a little bit of red off of one chromosome and a little bit of yellow off of the other chromosome, and we're literally going to have those trade places. That's known as a crossing over event. So we've got our homologous pairs partnered up in prophase 1. Think all the P's go together. And we've got crossing over occurring, which can increase genetic variation. You do in your picture want to color code this so that we can see that information has been exchanged. And now we're going to move on to the next step.